Our laboratory has found that hair follicle stem cells can not only reproduce the hair, but have a great multipotency to make many other kinds of cells, such as nerves. This presentation is going to be how hair follicle stem cells can be used for nerve regeneration and spinal cord regeneration. The hair follicle cycles throughout the lifetime of every mammal. The hair goes through a growing stage and a resting stage. When the hair follicle regenerates and produces a new hair shaft, the stem cells of the hair follicle regenerate the bottom part of the follicle. The stem cells of the follicle are located in a particular location in the hair follicle called the bulge. We have found in special mice that the stem cells of the hair follicle contain a stem cell protein called nestin. This very same protein is also found in nerve stem cells. In the special mice that we worked with, the gene for green fluorescent protein is linked to the nestin gene, so that in these mice, the hair follicle stem cells glow green when excited by blue light. We can see in this slide how the green fluorescent protein, or GFP, expressing hair follicle stem cells produce the hair follicle throughout the cycle of the follicle. This slide shows a higher magnification of the hair follicle stem cells located at the bulge area of the hair follicle. The hair follicle stem cells glow green here because the nestin stem cell protein that they contain is linked to the green fluorescent protein that has been engineered into special mice, such that every time the nestin protein is expressed, so is the green fluorescent protein. We can see the beautiful structure of these hair follicle stem cells, especially in A3, where we see many very interesting processes emanating from the bulb area of the cell. This slide shows the beginning of anagen, or the growth phase of the hair follicle. We can see the hair follicle stem cells expressing green fluorescent protein and glowing green start to form cells that are the beginning of the new hair follicle. The hair follicles are interconnected by structures that also express nestin, as we can see in this slide, since nestin drives the expression of green fluorescent protein. Every hair follicle seems interconnected at the area of their stem cells at the bulge location by these fluorescent structures, which we have later found to be blood vessels. When hair follicles are transplanted to other mice, they start to form blood vessels, as we can see in this slide. These blood vessels are projected into the skin where their hair follicle has been transplanted. The blood vessels express GFP, meaning that they also express the stem cell protein nestin. Hair follicle stem cells were isolated from the giant whisker or vibrissa hair follicle of the mouse. The isolated hair follicles were then put into a culture dish where they formed a sphere. The hair follicle stem cells in the sphere in culture continued to express nestin, since they were also expressing green fluorescent protein. When analyzed for stem cell related protein expression, we found that the growing spheres derived from the hair follicle stem cells express the gene CD34, which is also a stem cell protein. However, these stem cells did not express keratin 15, which is very important in the hair follicle, and also did not express the beta-3 tubulin protein, which is diagnostic of neurons, or the CD31 protein, which is diagnostic of endothelial cells that form blood vessels. However, when the hair follicle-derived spheres were put into differentiation medium that contained fetal bovine serum, they began to form neurons. We saw this because not only do the cells look like neurons, but they expressed beta-3 tubulin, which is diagnostic of neurons. The differentiating hair follicle stem cells in culture also formed keratin 15 expressing cells, which are diagnostic of keratinocytes that are present in the hair follicle. The hair follicle stem cells also formed astrocytes, since they expressed GFAP, which is a marker of astrocytes. Smooth muscle cells were also formed by the hair follicle stem cells in culture, since they expressed smooth muscle actin, a marker of smooth muscles. Melanocytes, the cells that form the pigment of the hair, were also formed from the hair follicle stem cells in culture. We could see that these melanocytes form the pigment granule containing melanin. We then looked at the percentage of cells formed by the hair follicle stem cells in culture. Neurons were most frequently formed, followed by glial cells and keratinocytes. Occasionally, smooth muscle cells and melanocytes were also formed.